Chattopadhyaya. So I request Surya sir to kindly conduct the session. Sir, please.
Thank you, Dr. Hussain, for giving me the opportunity to chair this session. So, good morning to all the participants. I am Professor Surya Chattopadhyaya, head of the Department of Physics, Tripura University, and it's my privilege to introduce Dr. Pabitra Kumar Pal on behalf of Department of Physics. Dr. Pabitra Kumar Pal is presently working as Associate Professor in Department of Physics, Jadavpur University, Kolkata. He has more than 16 years teaching and research experience. He formerly worked as Assistant Professor in Physics, both at our department, that is Tripura University, and the University of Bardhaman, West Bengal. He also worked as a visiting scientist at Osaka University, Japan, and at the University of Texas at El Paso as a Ramon postdoctoral research fellow. He is actively engaged in research in the area of material science, especially organic thin films and their various properties and applications, and chemical and biological sensors. Emphasis has been given to molecular spectroscopy and their applications in various restricted geometries in his research. He has published a large number of research papers in various national and international journals, and also participated various national and international conferences and symposiums. He also completed various research projects funded by state government, that is state government of West Bengal, and central government as principal investigator. He also served as a member of different academic bodies in various universities all over the India. So this is all about Dr. Pabitra Kumar Pal, and I am requesting Dr. Pal to present his lecture. Please, Dr. Pal, start your lecture, please.
Thank you, Professor Chattopadhyaya, for inviting me in this. Am I audible?
Yes,
Hello?
yes,
Okay.
yes.
Okay, so thank you, Dr. Professor Surya Chattopadhyaya, for inviting us here to deliver a talk about my research. And I also thank to Dr. Soed Ashad Hussain for giving me this opportunity to share my research experience in this FDP program, which is funded, affiliated by AICTE. So it's a very good morning, everybody. And today I would like to talk on a topic. I just share my screen. Okay. Is this visible? Okay.
Yes.
Okay. So today my topic of talk is the fluorescence resonance energy transfer, an efficient spectrofluorometric approach for various molecular level sensing applications. So before going to the main objective of this talk, I would like to just to discuss about the sensor, the basic idea of the sensor. What is sensor? Basically, sensor is a device used for the conversion of physical events or characteristics into electrical signals or any other detectable signals. So that we have for everyday experience, we face various type of sensors. That is one very easy example is the thermometer. Thermometer, when we measure the body temperature, thermometer basically it takes the temperature of physical characteristics from the environment and it converts into the electrical signal. That is, some material is inside the thermometer which is very much sensitive to the surrounding macroscopic environment. So that material is collecting the information, the macroscopic information from the environment, and it transmits as the input conditioning unit of the system. So this is the very simplest way of thinking about the sensor, and there are various intelligent sensor technology we use every day in our life, and for example ultrasound, non-contact radar, optoelectronic sensor, laser technologies, and fluorometric approach are the current trends in the industry, which drive the various innovations. So the next is the, as my topic is related on the molecular level sensing, so I think it is we should have some idea what is the molecular level sensing. So a molecular sensor or chemosensor, basically it is a molecular structure, maybe organic or inorganic complexes, which is used for sensing various analytes to produce a detectable change or a signal. That means it is basically a molecular structure which has the property to sense the analyte or to interact with the analytes, by which that molecule or that structure can modulate or can manipulate its physical or physicochemical properties, which can be converted into the detectable signal. So this is a very basic and very simple idea about the molecular sensing. And this type of sensing basically, or the molecular sensor or chemosensor, this basically relies on the interaction occurring at the molecular level. That's why it is called the molecular sensing. So, and it basically involves the continuous monitoring of the activity of the surrounding chemical species in a given matrix, such as it may be solution, blood, air, tissue, waste effluents, or drinking water, etc. So there are a variety of interactions, a variety of circumstances where this type of molecular interactions may happen. So, and this molecular sensor basically in general has the two type of thing, the mechanism. One is the signaling moiety, another is the recognition moiety. So, and these two moieties basically are connected together directly by some spacer. And the signaling, you see the picture here, the signaling moiety, this signaling moiety acts as a signal transducer, which can convert that information into the optical response in a clear and reproducible manner. So the signaling moiety is the active part of the molecular sensor, which basically collects the information by recognizing the target or analyte. And sometimes these two moieties are connected together. Sometimes it may not connect it directly. It may not connect with this moiety. So it depends upon the circumstances. So this is the very general idea about the molecular level sensing. And any perturbation or any any fluctuations or any change in the environment or the target analytes, basically it causes the changes in the physical properties of the molecules, of the sensing molecules. So ultimately in this process, the interaction with the optical signal, the electronic property, the photophysical properties, those are changes, and it is the change of these properties is many cases very linear, so that we can develop this type of molecular level sensing for the wide variety of the molecules or the target molecules or the environment, the macroscopic conditions, etc. So this is a very very easiest way to understand, to think about the molecular level sensing. And this molecular level sensing, as I already told you, that the optical signaling methods are very sensitive. These methods are involved in this type of the sensing, and this protocol or this strategy of sensing is very much sensitive and selective, and provide a platform for real-time response and local observations. And more interestingly, the chemical or the molecular level sensing is based on optical signaling, as I already told, and is suitable for monitoring real life events on cellular or extracellular level. So in our biological field or biophysiological field, that is this type of molecular sensing is very much beneficial, because it has the ability to interact with the cellular extracellular level very in an ultra fast and to ultra molar concentration. So that is why it's very much and also and additionally, this chemical and the molecular level sensing is very much selective to a particular bioanalytes for many biological, which is which is responsible for many biological processes in our body. So and also it is very easy to modulate because in the for the availability of the various optically active molecules, we can design various molecular level sensor which is which can detect a wide variety of molecular and even as well as biomolecular species. And various techniques are available to this type of to design this type of sensing platform. That is one is the Raman spectroscopy, fluorescence based sensor, fluorescent lighting imaging based sensor. So these are very common, and we are we use this type of techniques since long back, and there are a lot of evidences for successful application of these type of techniques to to design a various molecular level sensors. And the fluorescence in this presentation, basically our interest is to understand the fluorescence spectroscopic method to identify or to detect various physiological conditions, some macroscopic conditions, or some biological species of interest. So the fluorescence based, in general, this fluorescence based sensor, there are commonly three different type of sensing protocols are available. The first one is the fluorescence quenching, which is the fluorescence turn off sensor. The second one is the fluorescence enhancement and the fluorescence turn on sensor, and the third one is the fluorescence resonance energy transfer, which is of interest in this present today's talk. So the first one, the A, is the fluorescence quenching. Basically, fluorescence, as I already told you, fluorescence basically is a characteristic property of a molecule, and this is an intrinsic property, and this fluorescence intensity can be completely quenched in presence of any external agent or external guest particles or any macroscopic surrounding macroscopic conditions, etc., etc. So this and the fluorescence it is happened in this type of sensing. The optical signal, the fluorescence signal, is completely quenched, and the quenching of the signal is very much selective to the external target analytes or the external macroscopic conditions. So, and we can measure this type of fluorescence quenching by turn off by simple the steady state steady state fluorescence technology, which is available in our our scientific laboratories, and it's very easy to design this type of sensor. The second one is the fluorescence enhancement or turn on sensor. Normally we can design many molecules where some moieties, some groups are involved by which the intrinsic fluorescence can be quenched, or some complex we can design where the fluorescence is normally is quenched. But when this molecule is exposed to the external target bioanalytes or any other molecular species of interest to sense, the fluorescence signal is automatically come out and it is turn on. So this type of sensing is called the turn on, and both the turn off and turn on sensing is very much linear in many cases. So there are a lot of examples are available this type of sensor, and also this the process of sensing in this two way is very much ultra fast, because fluorescence is a process which involves the nanosecond time or time order nanosecond in this process. So detecting the molecular species is very fast. So both the two strategies are also helpful for designing many sensors for the molecular species, as well as the environment, the macroscopic environment. The third one is the fluorescence resonance energy transfer. I will talk to this topic later in this presentation, and that is our interest, fluorescence resonance energy transfer. And in this energy transfer, basically this is the fluorescence, and one the excited state energy can be transferred to a neighboring molecule, which can be regarded as a acceptor, and it's a non-radiative fashion, the transfer. So ultimately this transfer of energy is also a very much selective and sensitive. So that's why people or scientists have designed many sensors, especially biosensor and molecular sensor, based on this technique. And the another fourth approach, which is called sensing via intramolecular or intermolecular charge transfer in the for sensing the molecule, because this charge transfer may be photo-induced. So when the molecule, if we can design some molecule where there is a charge transfer, that the charge transfer can happen when we irradiate the molecule to the electromagnetic radiations. So, and this charge transfer can happen both in a single molecule, that is intramolecular charge transfer, or it can transfer the charge with the neighboring molecules or with the solvent. So so charge transfer, and you can see here it is a typical example for a particular sample. This is called KNP, for potassium to nitrophenol, and this molecule has a charge transfer property, and you see this is the charge transfer band, and it increases with the temperature 10 to 50 degree. So it is increases, so very selectively where it is sensitive to the temperature. So this can be a temperature sensor in this way. So it is a very simple strategy for utilizing the charge transfer between in the same molecule. It is the KNP molecule. So there are many other molecules you can design, or people already designed, scientists already designed many molecules which show this type of properties. So so as I told you, this is the fluorescence is our interest. So I think it is better to understand what is the fluorescence, and before going to the fluorescence, it is important to understand what is the electronically excited state. In this presentation, I will try to give you some very basics of the photophysical processes. I think some of you may be familiar with this topic. So I just try to recapitulate all these things, because this important discussions are very much helpful for to understand the resonance energy transfer, the FRET. So generally all kinds of luminescence are based on the so-called photophysical processes, and you know that the atoms of different elements have different number of distributed electron distributed in several shells or orbitals, and the electronic transitions are responsible for luminescence. So for the fluorescence, what I have told, this is also a luminescence. So the electronic transitions are responsible for the luminescence property of the molecule, and on absorption of the light, the electrons are excited and are lifted into higher energetic state. Before excitation, in the ground state, some of the electrons are in the so-called HOMO, or the highest occupied molecular orbital. You can see this picture, the right hand top corner, the yellow picture here. This is the ground state, and you see this is the highest occupied molecular orbital. So orbitals are occupied, and the upper this is the LUMO, is the lowest unoccupied molecular orbital. So after they reach the excited state, they are in the LUMO, and there is a LUMO, and this is called lowest unoccupied molecular orbital. So the transition, when the optical or irradiation happens, then the electrons from the HOMO to the LUMO level can jump, and after absorbing the energy, and electron can stay on the LUMO level for a certain short period of time, and then return back to the ground state. And I think here some of many of you may have idea about the molecular orbitals. So this is the molecular orbitals opposite to and contrast to the atomic orbitals. These orbitals are basically the linear combination of the atomic orbitals, And there are several type of molecular orbitals depending upon the energies, say n, sigma, pi. This type of molecular orbitals are commonly we use for our different photophysical processes, to explain different photophysical processes. So so this is the ground state, and this is in the excited state. What will happen? The one of the electron may one or many electrons may go to this excited state and then return back to the ground state. So this is an electronic state, the fluorescence. This is the I mean the transition may happen this type of in case of fluorescence of the between the same spin multiplicity. So there are different multiplicities for the possible electronic state possible. So singlet, triplet, etc. So in case of fluorescence, the same multiplicity transition between the same multiplicity will happen. So after photo excitation, and it is very fast, and the molecule can rest, or the electron can rest about 10 to the minus 15 seconds in the high excited state, and then return back to the ground state. So what are the then strategies of the deactivation of the electronically excited state of the molecules? So normally the electronically excited states are unstable, because it is the entropically it is not favorable, because it tries to return back to the ground state, because the potential energy is minimum. So as soon as the electron reaches the excited states, they try to return back to ground state because of the potential minimizations. And there are different different possibilities of exchanging the transmitting the energies when returning to the ground state. So excitation during the return to the ground state, the excitation energy is released again, and this release of excitation energy may happen in different way. It may happen in radiative process, it may happen in a non-radiative process. So in many molecules, those who are not luminescent, they can those molecules lost the energy, the excitation energy non-radiatively to the surrounding molecules or to the environments, or the excited energies can be lost during the collisions when returning to the ground state. So there are several ways to lose the excited state energy while coming to the ground state. So as I told you that both radiative and non-radiative decay processes may happen, and most of the time the decay is non-radiative. For example, this non-radiative means the vibrational relaxations, quenching with the surrounding molecules. That is, I have already told that collisions during the molecule with the other rest of the molecules, or internal conversion. So I will come to the point internal conversion and intersystem crossing, etc. So this is the schematic diagram of this is called Jablonski diagram, where this is the this represents the pathways of the activation of the deactivation of the electronically excited molecules. So the deactivation pathways, there are two ways: the radiative and non-radiative. The examples for the radiative transitions or radiative deactivation is the fluorescence, phosphorescence, etc. And the non-radiative processes are it includes the internal conversion, intersystem crossing, etc. In case of the radiative transitions, the emitted light has a longer wavelength and a lower energy than the absorbed light, because a part of energy has already been released in a non-radiative decay process. That means it's very easy to understand that when the molecules excited, so the molecules, the molecules have different degrees of freedom. So translational, vibrational, rotational, etc. So when there is a irradiation of the molecules with the electromagnetic radiation, the molecule can vibrate also. So their vibration energy can also change during the photo excitation, and when it returns to the ground state, then the molecule or the electron has to pass to many different vibrational levels, or sometimes it can cross, the molecule can overlap with or interact with the vibrational levels of the neighboring molecules. So there is many possibilities of losing the excited energy. So not all the energies will be utilized to emit the radiation. So all the absorbed energies will be utilized to emit the radiation. So that is why the wavelength of the emission of the light is much higher than it's much not or much, it is always higher than the absorption of the light. So and energy is wavelength is higher means energy is less. So it is very obvious because of the loss of energy. And sometimes although it is the contribution is very less, that sometimes some of the energies can be distributed to the neighboring environment, to the surrounding environments in the form of the heat. But this process is many cases the contribution is very less. And during the deactivation or after the deactivation, we can if we monitor the intensity of the emitted light, we can check, we can see that there is a shift from the absorption peak to the emitted emission peak. So there's the difference between the absorption peak and the emission peak is known as the Stokes shift, and this is very much important to design to various photophysical to understand various photophysical processes and to design various the molecular sensors by using this Stokes shift. So, but ideally, if you talk about a single molecule, if there is no perturbation, no interaction with other molecules, no interaction with the solvent, the molecule is in a gaseous state and isolated molecule. So ideally it should be a mirror image relationship between the absorption and emission. But in real practice, there are several other factors involved which interferes with the emission of the radiation. That's why there is a difference between the two. So you see that this picture, the Jablonski diagram, if the initially electrons in the ground state S0, and when after photoexcitation the electrons will reach to the S2 state or S3 state, whatever. I have drawn here only the two states, S3 state, S0, S1 and S2. And after photoexcitation, you can see what I have already explained here. The electrons can cross this vibrational level, and then then the electron can ultimately to reach to the S1 level, and this changing the states of the electron is called the internal conversion. And again, the electron can vibrationally relax to lose the energy to reach the zero vibration level of the S1 state. And then you see the green lines here from the zero vibration level ultimately to the excited electron can reach to the zero vibrational state of the ground state. But the electron can, all the states are probable. So electron can reach to the any of the vibration state of the ground state, but finally at the zero vibration state the electron can reach. And as you know, this process is also the temperature dependent, because the population of the electrons in the ground state or any electronic states basically is governed by the Boltzmann factor e⁻e by kT. Sometimes during the photoexcitation it may happen for some samples, for heavier samples where the spin orbit interactions is much more prominent, then the electrons can cross to the this is the another electronic state which is called triplet state. It is for different multiplicity, because the spin orientations are parallel here, so and here anti-parallel, so spin has to flip to go to the T1 state. So and ultimately also the molecule vibration relax to the zero vibrational state of the triplet state, and then come to the ground state of the electron. And this process takes much more longer time compared to this fluorescence. So that's why this is called the delayed emission, and this is called the fluorescence. So the lifetime of this type of emission is much more longer compared to the fluorescence. So that means from this diagram you can have an idea about the transition and the rate of transition. And one thing is very important to emphasize in this presentation, that these transitions are spin allowed transitions because spin multiplicities are involved. So these are called spin allowed transitions. And you can find this type of things in many MSc master degree courses. So this may be, I think, very, very helpful for our general knowledge for the molecular spectroscopy and those who are working with molecular spectroscopy in their research. So I have already told about the what is fluorescence, because fluorescence, it is very important to understand the fluorescence, and this involves the electronic transition between like spin multiplicity, already told you, and the emission has a short lifetime, that is 10 to the minus 2 to the minus 5 second, and usually takes place from the S1 level after photo excitation. But for the condensed system, that means for the liquid solution or the some solid sample or solid matrix, the excited vibrational state populations quickly relax to the zero vibration level of the excited state due to collisional interactions or the exchange of photons. That means the different electrons can be excited to reach the excited states, and that photons also can interact with the electrons. That means there will be many possibilities of exchanging the or transferring the or losing the absorbed photon in different forms or different way. So, and as I've already told you about the Stokes shift, you can see this picture that this is the Stokes shift, this absorption peak and the emission peak. So blue light, and this is the green fluorescence. So this is this may happen almost in every sample radiating the fluorescence or emitting the fluorescence radiation. So and this is the single molecule fluorescence, but as I told you, this is not practically happen because practically the molecules we do experiment in reality for many molecules, and molecules are there are various interactions involved. So practically this is not possible, but this is a very general to understand the fluorescence, we can think in this way. So this is in most cases the absorption into higher excited states Sn is followed by relaxation into fast singular state, and the emission is occurred due to the transition from zero vibration level S1 to zero or higher vibration level of the ground electronic state S1. And this is a very important statement, and this is the very famous Casas rule, and which is we should know when we talk about the molecular spectroscopy, especially the photophysical processes of the electronically excited molecules. So this is very interesting rule. So before going to the fluorescence resonance energy transfer, I repeat once again, that is the photophysical processes. This involved various type of interactions between the molecules in reality, and radiative and non-radiative processes are involved. Not entire absorption will be the radiative. Some of the radiations will be non-radiative, and some of the will be radiative. So whenever you talk about the interaction of the molecules with the other guest molecules or the any environment or the molecules present in the environment or the medium where our molecules we can think that the transfer of energy there, what is the way of transfer of energy. So I have already discussed some of the possibilities. Now in this presentation I would try to explain about the resonance energy transfer. What is the resonance energy transfer? In what way the energy can transfer from one excited molecule to the other neighboring molecules? So that is the fluorescence resonance energy transfer. And this this fluorescence resonance energy transfer is basically widely used as a spectroscopic technique in various applications of fluorescence, including the medical diagnostics, DNA analysis, optical imaging, and for various sensing properties. And this, I've already told you that fluorescence based sensors adopt 3 different strategies: turn on, turn off, and the energy transfer. So here we are talking about the fluorescence resonance transfer, and this FRET sensor become popular tools for studying intracellular processes. I mean, because our cell has different type of organic and biological molecules, some ions, some proteins, some lipids, uh, or some antibodies, some enzymes, or some different type of biological species may present, and and their interactions, their activity basically activity implies basic various photophysical various biological processes for our body. So the FRET is an interesting technique, and it can be used uh, fantastically, or it can be used very accurately to understand the biological process. We can this technique can very easily recognize the biological phenomena, or the present of the various biological components, the amount of various biological components, the organic components, inorganic components, ions, etc. You can easily understand by using the FRET. So it has the potential applications in the biological field, the medical industry, and also the potential applications in optoelectronic and thin film device development. So because nowadays the FRET is also observed in solid state also, so that's why this technique is also important for the thin film device development. So, and we'll try to, in this presentation, we'll try to understand some FRET sensor, especially some biosensing and hard water sensing, etc. So the scientists have developed many FRET sensor, and this is not new, but this is very interesting and promising, because lot of possibilities are there using the FRET technology, because FRET is basically a ratiometric sensor, because the presence of the perturbation of the external environment automatically cancel out this in this type of sensing technology. So I just briefly try to explain, because time is short, so I try to explain this, what is the FRET actually. So after photoexcitation, the excited donor flow, there are two fluorophores are involved, two type of fluorophores. One is called donor, another is called acceptor, and both can emit radiation independently. But when the situation is something like that, when the excited donor fluorophores transfer its energy, excitation energy, to the neighboring acceptor chromophore in a non-radiative way via the long-range dipole-dipole interaction. So we can see some quenching of the fluorescence of the donor molecule, and this quenching of the fluorescence of the donor molecule is very systematic, with increase of the concentration of the acceptor molecules. The mechanism is that the two molecules, donor and acceptor, after photoexcitation, only if I excite the donor molecules only, so donor molecules dipole, the dipole can oscillate, and when due to the vibrational reactions, it can reach to this S1 level, as I already explained in our previous slide, and instead of returning to the ground state, this vibrational the absorbed energy can transfer non-radiatively to the neighboring molecule in the excited state. That means this energy, the resultant absorbed energy after the vibrational relaxation, the resultant energy can transfer to the neighboring molecule, and the neighboring acceptor molecule I am talking about, and that molecule can oscillate resonantly with the donor molecule. That means you can think this oscillation in this way, you can think the two tuning forks are vibrating in the same frequency. So that is in case of the coupled oscillator. So the two molecules, now they are vibrating with same frequency, so their transitional dipole moment they interact with each other, they couple together, and ultimately the energy is transferred to the this acceptor molecule, and by a natural process, it is the intrinsic property of the acceptor. But energy is high, so it has to relax through this vibrational relaxation, and then come to the ground state. So in this way, this acceptor molecule can emit after getting photo excitation from the excited donor molecules. So it is very interesting, very very easy to understand this mechanism. It is not difficult, and but there are some conditions required for this to happen. So, and this excited the molecule one thing is very important, I forgot to mention, that this excitation actually this should have a this acceptor molecule and this donor molecule. The excitation of the acceptor molecule and the excitation of the donor molecule should have some far away from the donor molecule. The excitation of the acceptor should be away from the excitation of the donor molecule, so that when we excite the donor molecules, we can avoid the direct excitation of the acceptor molecule. That is, choosing the two molecules is very important. That is, we need to keep in our mind that what is the excitation wavelength for both the acceptor and the donor. So that is very crucial. And Stokes shift, also the Stokes shift is also important for the donor molecules, because the emission of this large Stokes shift is required, because otherwise it cannot excite the acceptor molecules, because excitation is large very much different compared to the donor molecules. So then what are the main criteria or conditions for this fate to happen? You can see this picture here. This is the absorption spectra and the emission spectra. This blue one, these two, the yellow. So this one and the red one is the absorption and emission spectra of the acceptor molecule, that is donor molecule, and this is the absorption and emission spectra of the acceptor molecule. So the main condition is that there should be sufficient spectral overlap between the absorption spectra of the acceptor and emission spectra of the donor. So the green shaded region is, you see, this is the spectral overlap. From this picture, this diagram, it is very much obvious this is required. Otherwise, there is no chance to transfer this energy, because this is the resonant interaction. So somehow this emission and this absorption, there should be some overlapping between the two. So that means the degree of overlap is referred to as the spectral overlap integral. If J increases, so the energy transfer increases. The second one is the two fluorophores must be in close proximity to one another. That is, the molecules, depending upon the concentrations of the acceptor and the donor, the distances between the concentration, donor and acceptor should be always 1 to 10 nanometer. That means 10 to 100 angstrom. Otherwise, collision will occur. If we increase the much more concentrations of the solution, then collisional quenching will be there, and which and that is also the non-radiative process, and it can also hamper or affect adversely on the fluorescent energy transfer. But concentration is important, because number of molecules are required to get the to increase the fate, because when you measure the fate in the steady state, the intensity comes from the number of populations of the molecule in the excited state. So if we have much more number of molecules in the excited state, so there is a much more possibilities to transfer the energies in a larger amount. So that is when you integrate all the photons coming from all the due to the all the interactions involving in the fate process, so automatically the fate will be increased. But there should be some limit, otherwise the collisional quenching will occur. and freight will diminish. And the transition dipole moments of the donor and acceptor must be approximately parallel to each other. So the transition dipole moments is the when the molecule, everybody we know what is the transition dipole moment, and because we have an idea from our master degree knowledge, the perturbation theory, and when the molecule excited irradiate with the external radiation, then dipole will oscillate with the external radiation, and ultimately the resultant transition dipole moment will give the molecule for the emission of radiation. So the transition dipole moment is basically the two transition dipole moment, two acceptor molecules must be parallel to each other. That means 0 degree, because the angle between the two should be 0, because it is the dot product. So cos theta cos 0 is 1, so that is a maximum, and if it is perpendicular it is minimum, because cos 90 is 0. So fluorescence lifetime of the donor molecules must be of sufficient duration to allow the freight to occur. So if we have the molecules of very low lifetime, that means the molecule will after photoxide it will very quickly relax to the ground state. So there will be no opportunity to transfer the energy to the neighboring molecules. Or it may, you can consider that the very small amount of energy may be transferred. So the energy extent of energy transfer will be not sufficient for any detectable signal or for the practical purposes. Well, one parameter is very important during the freight study or designing the freight sensor, and that is the freight efficiency, which is very much important to understand. The freight efficiency is defined as R0 to the power 6 divided by R0 to the power 6 plus small r0 to the power 6. Here basically capital R0 is the Froster radius at which the half of the excitation energy of the donor is transferred to the acceptor chromophore. That is 50% of energy should be transferred to the donor molecule, so that the energy transfer is 50%. And this is basically measured by the steady state measurements, steady state fluorescence measurement. You can easily measure the steady state intensity, fluorescence intensity, and you can calculate this by using the by measuring the simply the intensity of the emitted radiation from the donor molecules only. So when the 50%, then the distance at which this is happened, this is called the Froster radius, and the rate of this non-radiative energy transfer according to Froster, because this was first designed by Froster, so and that's why it's called Froster. That is why it is also known as the Froster resonance energy transfer. And K tau is the KT is the Froster resonance energy transfer, and this is given by this equation 1 by tau r0 to the power 6 divided by r. And what is the small r? Small r is the distance, instantaneous distance between the donor and acceptor molecules, or any at any time. And KT is the energy transfer rate, and tau d is the fluorescence lifetime of the donor, and r is the distance between the donor acceptor, as I told you already, for a particular donor acceptor pair at any given conditions. And this Froster radius is also defined by the following relation, and there are theories available in the literature. You can also go through the theory of the freight to know much, to have some idea about this theory. And FD, here this equation, you see the FD, and FD is the normalized fluorescence intensity of the donor molecules at any particular concentration for which freight is occurred, and phi d is the quantum yield for the of the donor. The amount of energy transfer also depends on the extent of spectral overlap, which is basically the integral part of the equation. This J lambda is the spectral overlap, as I already mentioned in our previous slide, the spectral overlap, the green color shaded region. So this is the basically the area under the curve, because area under the overlap region of the curve. So this the 0 to infinity FD and epsilon A, and this is the extension coefficient, and this is the lambda to the power 4, is the wavelength. So therefore the expression R0, we can using these two equations, we can write R0, the Froster radius, in this way. And considering all these equations, we can also write the FRET efficiency as E is equal to 1 minus FDA by FD. Here FDA, the numerator, is basically the steady state fluorescence intensity of the donor in presence of acceptor, and the denominator FD is the fluorescence steady state fluorescence intensity in absence of the acceptor. So we can by considering this ratio and by this equation, one can into 100%, if you do, we can express the efficiency as a percentage. So and so it cannot exceed the 100%. So any process that affects the energy transfer rate allows the process to be quantified. So that means by this equation one can easily quantify the process, I mean the interactions you can quantify. So and this is in many cases this energy transfer efficiency is very much linear with respect to the interactions, with respect to the interaction with the guest or external molecules or external or the surrounding microscopic environment. So that's why scientists have developed this theory, they developed this technique to design various sensors. And one thing very important, you see, this is the ratiometric sensor. So when you take this intensity, the ratio of these two intensity, so if there is any perturbation or any fluctuations present in the studied medium, so it will be automatically cancelled out, because fluctuations will be for the both the donor and the acceptor. So you can easily cancel out those fluctuations associated with the medium or with your measurement, or fluctuations in the temperatures, anything, all these things can be automatically cancel out by using this equation. So the FRET, that is why FRET is very much efficient and accurate measurement for determining the molecular species or wide variety of molecular species from the and to a very ultramolar concentration. So that's why we use the FRET for different sensing. That is our interest in this present topic, in this talk, the FRET, fluorescence energy transfer, how to design this energy transfer, how to use this technique to design various sensors. Now, how to detect FRET? So you have the instrument, you have this all these materials, the donor and acceptors you have chosen. So you can choosing the appropriate FRET pair molecules by considering all the basic things what I have explained, and choosing the appropriate FRET pair molecules and checking their absorption and emission profile of the both fluorophore, and choosing the right excitation wavelength. Because I just explained here, because choosing the right excitation, the molecule can excite in different at different wavelengths. Only the difference is that there are many molecules had different excitation wavelength, and but only difference is that the emitted intensity is changing, because the number of molecules for different wavelengths, the number actually changes. That means population in the excited state can vary when you change the excitation wavelength. So that's why there is a different excitation wavelength for a particular molecule, but you need to choose the particular wavelength so that wavelength cannot directly excite the acceptor molecule. So this is very important, we should keep in your mind while designing the FRET pair and for a sensor to design the sensor. And direct excitation of the acceptor molecule must be avoided, I have already explained. In solution, the spectroscopic titration is to be carried out for mixed dye pair or mixed fluorophore pair by increasing the concentration of the acceptor molecules, keeping the concentration of the donor fixed. So in this picture you see that this is the donor, the pure donor, fluorescence of pure donor, and the blue is the fluorescence of pure acceptor. but it's very much less compared to, I mean, negligibly small compared to the donor. So, and the same excitation wavelength. And this keeping the, so we can fix the concentration of the this donor, and we can increase the concentration of the, I mean, the acceptor. And you see this decreases, and this band in the mixture, this band decreases and this band increases, and this band is for the acceptor band. So acceptor band will increase, and the intensity of the donor band will, emission band will decrease systematically with increase in the concentration of the acceptors. And effect of any external guest molecules or change in any macroscopic condition can be checked. So we need to also check the what is the effect of external gate molecules of interest, because if you want to design some sensors, or if you know some possibilities of interactions, selective interactions with the any guest molecule, then you can easily design this the sensor. And or for every fluorescence spectra of the mixed dye solution, the FRET efficiency is calculated. So for every fluorescence spectra, I mean the mixed solutions, you can efficiency of the for quenching, every quenching you can calculate by using the equation of the efficiency. And this is the typical instrument which you can use. This is from Horiba instrument, the steady state fluorescence spectrophotometer. So, and it is available in many research laboratories. So it is very easy to work in this field to explore various possibilities in the area of molecular sensors. So up to this, basically I have explained some of the basics about the photophysical processes, basics about the molecular excitation or electronic excitations, basics about the FRET technique, their conditions, etc. And also the what is the sensor, the general consideration of sensors, and also what is the interesting benefits of FRET technology to design the sensors. So all this we should keep in your mind while talking, while we can proceed for interesting work for the sensor. So these are very prerequisite understanding which everybody should have to proceed for the for utility of the FRET to design various sensors. So then FRET is a choice for various molecular and biomolecular sensing. So FRET has been employed to recognize various selectively various molecular and biomolecular species from a wide variety of conditions. And some recent research suggests that successful design of efficient platform ion sensing, pH sensing, temperature sensing, the biological sensing, etc. So there are several works, several literatures available in the internet or in the journals. You can find many different interesting work. So I can here just highlight some of the works which we did and using the FRET technology for measuring the to sense the target molecules or the environment, macroscopic environment, etc. And one thing I should mention, that is one can directly use FRET efficiency, and one can indirectly use the FRET efficiency or FRET technology to measure to selectively detect the various molecular or biomolecular species. So there are different approach of thinking or utilizing the FRET technique or explaining the sensing protocol in different way using the same using the FRET technology. So the work what I would like to mention here, that is a selective and sensitive detection of L-Cysteine using gold nanoparticles and rhodomine B in aqueous solution. L-Cysteine is a, so this is an example of that FRET is involved in this type of measurement actually, and selective and sensitive detection of L-Cysteine using gold nanoparticles. And everybody here, we are concerned about the gold nanoparticles, and lot of examples are there. A lot of utilities of gold nanoparticles you may know. So, and rhodamine B is a cationic dye, and it has used application especially in the biological and field. And L-Cysteine is the sulfur containing amino acid and is naturally derived in our plasma, is the building block of proteins and involved in many biological processes. And this amino acid is widely used in pharmaceutical industry as medicine to treat different type of physiological complications. That means deficiency of this amino acid is important. Harmful for our body, and it can causes some disease like neurotoxicity, promotes urinary stone, and the elevated amount, the excess amount is not good for our health, but it is also the building block for the protein. So for healthy adults, about 4.1 kg milligram per kg per day it is required for our human adults, and it is recommended by this expert committee. And in this work, basically in this work, this is the structure of the L-Cysteine, the molecular structure. You see there is a amine group, and the thiol group is over here, and hydroxyl group is here. So it is water soluble definitely from this structure. And I will not go to the detail of this work. Actually, I just highlight some of the results of this interesting work. For gold nanoparticles in aqueous colloidal solution, it has the band 528 nanometer, and it corresponds strong characteristic surface plasmon band for both longitudinal and transverse surface plasmon resonance. And there are two weak humps also. They are 251 and 356. You see this picture, and correspond to intra-band electronic transition. And from this picture, you see that there is a sufficient spectral overlap between these two. This is the acceptor gold nanoparticles, and the absorption spectra of gold nanoparticles and emission spectra of the rhodamine B. It has a large spectral overlap, and this is the tame picture. You can see the structure of the gold nanoparticles, and fluorescence of the dye molecules quenched when after addition of the gold nanoparticles to the dye solution. The fluorescence, you can see this picture, this one. The fluorescence. Am I audible, everybody? Hello?
Yes.
Yes, you are audible.
Okay, thank you. So, so this is the you can see this fluorescence is completely turned off here almost when you add the rhodamine B, gold nanoparticle to the solution, and when we add the L-Cysteine solution in the mixed solution, then the fluorescence is come out or is turned off. So initially the fluorescence is quenched by the Fred process from the dye molecules to the excited electrons. Energy can be transferred to the gold nanoparticles because it has large overlap, and this is fluorescence turn on. So, and this fluorescence recovery of the fluorescence is very much selective to this particular amino acid, and the amount of the recovery of the fluorescence is basically linear to the concentration of the analytes L-Cysteine. That is, we can think in this way: the separation between the gold nanoparticles and the rhodamine B is basically changing with respect to the when we add the L-Cysteine, because L-Cysteine has a sulfur containing amino acid. It can interact with the gold nanoparticle very strong through the thiol moieties or thiol group. So effectively the rhodamine B molecules can be separated, can be far away from the gold nanoparticles. So there is a less interaction with the less possibilities of interaction with the gold nanoparticles for the rhodamine B. So intrinsic fluorescence will come out. So and this is very selective and very systematic change, so we can have this fluorescence signal, and we can measure the signal. We can find out the concentration of the unknown amino acid, L-Cysteine from the mixture. So, and this is the protocol, schematic protocol presentation of the recovery of the fluorescence, because the energy transfer is basically modulated under the application when we apply the L-Cysteine. So, and this modulation and this change of the energy transfer is basically linear. That's why you see this curve. This curve is linear. The log 60 concentration, and this is linear. So that's why we can talk about that. Is a very good platform for sensing the amino acid. How much time I have, Dr. Hussain, please? Hello.
15
Hello?
minutes. 15 minutes. 15 minutes.
10 minutes.
15.
Okay. Okay. Thank you, Dr. Hussain. So this is the fluorescence of rhodamine B, and a strong interaction with L-Cysteine and AU nanoparticles through the thiol moieties facilitates separation of rhodamine B molecules from the AU surface, decreasing the FET quenching. So the quenched, what I explained, that is the increasing the concentration and quenching is basically decreases. So, and we also check the control. We have checked the control experiment. That is the fluorescence emission of this mixture, rhodamine B and the L-Cysteine mixed across solution without gold nanoparticles, reveals a negligible effect of L-Cysteine on the emission profile of rhodamine B molecule. That is, without the gold nanoparticles, there is no change of the emission property of the gold rhodamine B molecule. So, in this process, this nanoparticle has a fantastic, utility or significant interesting utility to measure the concentration of the L-Cysteine. And we plotted here the fluorescence recovery efficiency at 74 nanometer. This is the emission of the rhodamine B, and we have designed a calibration curve this way. And this is the steady state. From the steady state measurement, you can easily calibrate this. Easily calibrate the sensor in this way by this equation. So from this equation is a Statel equation you see here. So it's very much linear, and in our studied experiment, in our observations, we found that the limit of detection of this sample is about 0.01 micromolar. So this is the sensibility of the LOD. The limit of detection is called the sensibility of the LOD of the proposed L-Cysteine detection method can be comparable with the results reported in the literature. So, and this work is already published in a journal already, and we have also checked the selectivity of the proposed method. So we can check various other essential and non-essential amino acids present in our blood plasma, and you can see here only. In case of you can see the blue one here. This second top right corner, this curve. So this is L-Cysteine and all other amino acids. There is no significant detectable signal compared to this one. And we can check the interference of the other essential and non-essential amino acids. And we also found the similar result, that is presence of L-Cysteine gives the recovery of the signal. That's why it's a L-Cysteine. So selectively is detected L-Cysteine. And we applied this the technique for the real biosample, the human urine, and because the amount of a large amount of cysteine is excreted, the excess amount of cysteine is excreted through our urine, our excretion due to excretion, and you see the urine plus L-Cysteine is like this. So the blue one, and normally this is the normal urine. It also has the cysteine, but if we add this L-Cysteine, it increases. So, and if we add other amino acid to the urine, there is no change. There is no increase of the fluorescence intensity. So that means in real sample also this technique is works well. So it's a very good application for to designing a biosensor using the FRET is also the principle of FRET is involved here. So that is fantastic. That is very beautifully manifested here to design the FRET sensor for to detect the L-Cysteine from the variety of the other amino acids in the mixture. Next, biosensor, what we did in collaboration with Dr. Hussain. So the sensing of DNA conformation based on change in FRET efficiency between laser dyes, and this is published in Sensor Actuators B. And in this work, basically we focuses on the changes in FRET efficiencies between two dyes, acrylflavin and rhodamin B, associated with LVL films. So basically here we studied the FRET in the solid state, that is in LBL films, in presence of double-stranded DNA as well as a denatured DNA. So denaturation of DNA has been studied in a wide range of changes of pH and temperature, because you know the temperature and pH change in temperature and pH can alter the structure of the deoxyribonucleic acid, and DNA is the primitive building block of our living cell. So it is important to understand this to the structure of the DNA. So the FRET technique is used here to determine this structural information of this DNA. So, and this technique is basically potential application in optoelectronic and thin film devices, and also important for better understanding of some biological systems. So here I can just briefly explain that. So this highlighting some results. So this is the fluorescence spectra of acrylflavin and rhodamin B mixture, and solution in LVL films. So, and this is the technique of preparing the LVL film, the alternately the polycation and the anionic substance deposited in a solid substrate, and we have measured the DNA, the absorption. You know, the DNA has absorption intensity of 260 peak. So this is the variation in absorption intensity with respect to the pH, because we have changed the pH of the DNA solutions, and we see that this is maximum. And we studied when we prepared the LBL film of acrylflavin and rhodamin B and exposed to the DNA solutions at different pH, and we find that, and we also obtained the energy transfer efficiencies, and we found this type of plot. Okay, so energy transfer efficiency is minimum about 13 something like that, 13 pH about. So you see that the presence of DNA increases the FRET efficiency between the two laser dyes activated LVL film, because DNA, when we add DNA in the presence of two dye, so it eventually decreases the distance between the dye molecules. So, and the denaturation, and also increase in pH or temperature results in denaturation of the denaturation followed by the coil conformation of DNA. So at very high temperature, 95 degrees centigrade, and high pH about 13 or something like that, so DNA the hydrogen bond can break between the base pairs, and ultimately the DNA is denatured and it becomes single stranded, and finally it becomes coil-like structure because of the entropically that is the entropically favorable structure for the DNA at that particular condition. So the denaturation of the DNA leading to increase in intermolecular separation between the activated rhodamin B, and that's why the FRET efficiency increase. So here you see this curve, the temperature versus FRET efficiency. The after denaturation, the FRET efficiency increases. So at this portion, if we monitor this observation in some situation, we can have the information about the structure of the DNA. So coil conformation of the DNA strands marginally decreases the intermolecular distances between the acrylflavin and molecules, resulting in corresponding increase in FRET efficiency. So this is another example for how FRET is beautifully used to understand the conformation of the DNA. So observing the change in FRET efficiency between two laser dyes attached to the DNA strands, it would be possible to detect the altered DNA conformation in the changed environment. And this is the the atomic force microscopy. And this is the the normal ambient condition, the LVL film. and this is high pH and low pH. Sorry, high temperature at pH 13 and pH and temperature 95. And you see this picture, that this you can see that separation, the maximum Fred, because they are very close to each other, that separation, and minimum Fred because the separation basically maximum separation. And here Fred increases with coiled DNA and separates with the decreases compared to this one. So, and this is the another biosensor. The first one is also a biosensor, second one is also biosensor, and third one is also Fred based hard water sensor. The mineral content or the water, basically there are some minerals are present. The hard water is basically high compared to soft water, high compared to soft water, though the hard water is not harmful to one's health generally, but can cause serious problems in the industrial settings where water hardness should be monitored to avoid breakdowns of the costly equipments that handle water. So normally the most common cations which present in the hard water is calcium and magnesium. So, and also dissolved carbonate minerals, calcium carbonate and magnesium carbonate also provides some temporary hardness to the water. So we can also use the Fred technique to understand what amount of what is the hardness of the water, what amount of calcium and the magnesium ions are present in the water, so that we can have an idea about the quality of the water. So in this, it is very important to identify the permanent hardness of the water before use. So this is we use this technique. I just show you some results. So this is the concentration of the magnesium and ions, and we find that the Fred efficiency increases about 43.2 here, and for soft water is basically fluorescence spectra. I can explain just here. The fluorescence spectra of active pigment, here the same dye as before, the active pigment rhodomin B. In clay, we used clay dispersion also here with the calcium chloride and magnesium chloride of concentration 0.05 milligram per milliliter and 0.06 mg. So different concentrations we have considered, and we show that this is the concentration versus Fred efficiency, and this is the same thing. Here we find that for hard water it basically about 13.5% the efficiency, and the moderately hard water it becomes 48.2, and for soft water is very high, about very close to 70% of the energy transfer. So by monitoring this energy transfer, one can easily have an idea about the hardness of the water. And this is the schematics for the distances or separation between the molecules in presence of the ions, by which we can have an idea about the process of the energy transfer during the investigation of the hard water. So, and future optical methods for so up to this we have used the fluorescence technique for the collection of molecules, a group of molecules, or in real molecular systems. They were or in real bulk molecular systems, or sometimes in restricted geometries on thin films. But in the coming days, the optical method will be for the single molecule counts, because single molecule here, it is very interesting to mention that it can have the digital properties, like the single molecule has some intrinsic the fluorescence, and if there is no molecule, so no fluorescence. If molecule, the fluorescence will be there. So the signal will be very digital, so 0 or 1, something like that. So by using the single molecule technology, fluorescence technology, one can understand one can modulate the signal, one can manipulate the signal, or one can process the optical signal digitally for better accuracy and better understanding of the, or better picture of the sensing mechanism. So this is the single molecule technology. So scientists are devoted their efforts to in this area. We hope that coming days this will be much more helpful for to design the single molecular sensor for better understanding, for better clarity of the result and ease of processing, etc. So in conclusion, I just mentioned few points that fluorescent resonance energy transfer is an excellent choice for designing sensing platform for various molecular and biomolecular species, as well as to understand various macroscopic conditions surrounding the molecular environment. FRET is an ultrafast photophysical process where excited state energy transfer non-radiatively to neighboring acceptor molecules whose excited dipole oscillations is in resonance with that of the donor molecules. Ability of the variety of fluorophores make this technique a promising choice for recognition of various cellular or extracellular behavior with ease and minimum experimental sophistications. As in FRET, the estimation of efficiency is ratiometric. I'm sorry, the spelling is mistake. This is the ratio. It is to be the ratiometric. So the effect or any external perturbation automatically cancelled out, giving most accurate result. So you can try with the FRET technology. Those who are working in the spectroscopy or material science, this is very interesting, and a lot of possibilities and opportunities are there in future. So we can explore much more in this area to design new sensing platform for and with which will be very cost effective, cheaper and easily available and easily accessible to the the society. So I thank you so much, the organizer and the and especially Dr. Hussain and all the audience and participants, distinguished scientists, researchers. I thank you all for listening, for your patience to listen this my talk, and I hope you understand my talk. Thank you very much.
Thank you, Dr. Pal, for your nice presentation. Now, I'm observing that there are some questions from participants. I'm starting with Dr. Rangaraj Bhattacharya. He asked, kindly explain the meaning of potential minimization in case of deactivation.
Oh, actually, yes. I just a minute. So potential, actually, thing is that when excited state, the molecule is in excited state. So potential energy, the electron has to overcome various interactions, and the potential energy is basically increased. So but it is the natural tendency of the molecule, because molecule vibration, when the molecule is related with the optical signal or electron radiations, so the molecule also vibrate. So vibration energy also change. So overall the total energy is changed, and the molecule has to overcome the interactions with other molecules, the interaction with the medium, etc. So potential energy should be should increase. So but it is the natural process to deactivate, to go to the minimum potential by releasing the but to reach the minimum potential configuration of the molecule. So there are different way to this laws to minimization process. That is, the non-radiative process will be there, vibration relaxation will be there. The molecule can resonantly transfer the energy to other neighboring molecules. So there are many different possibilities, and to return to the ground state. Thank you, Dr.
Okay. Well, another question from Dr. Siddharth Bharadwaj. He asked how to ensure excitation energy given to donor does not affect the acceptor.
Okay, it's very simple, because we can initially check the excitation of the we can first check the absorption spectra of the both the donor and the acceptor. Okay, and we can find out the maximum absorption wavelength and for both the donor and acceptor, and we can check the emission spectrum for the both, and finally we can check the mixture of the two samples, and we can excite the donor excitation wavelength, that is the maximum absorption. Generally we can choose the maximum absorption wavelength but what happened? We need to sometimes we can compromise with the maximum excitation, because sometimes it can interact with the acceptor also. So that's why we can compromise that excitation to some lower wavelength, so that to avoid the direct excitation. But we need to choose the sample in such a way so that there is a large Stokes shift between the yes.
Well,
Yes.
actually, another question from Dr. Omid Gupta.
Okay.
He asked me, what is freight membrane?
Oh, that is actually I didn't work on that area. Plate membrane is basically some maybe some biological membrane where some the molecular interactions, some biological. So I didn't work on this area, so I can go through some literature, okay, to this thing, okay.
Well, actually, actually, Dr. Siddharth Bharadwaj asked another question. Is there any standard mixing ratio for the freight pairs?
Oh yeah. Yes. That is yes. Basically, there is no standard. Actually, thing is it depends upon the system of under interest. So that is different system had different concentrations. Okay, to work, because the separation between the donor acceptor depends upon the concentration, but the molecular size and their interactions is different for different samples. But there will be some optimum concentration for which the freight is maximum for a particular pair of donors and the acceptors. So it depends.
Okay. So another question from Dr. Bharadwaj. Is there a chance of photo bleaching between dye pairs?
Yes. Actually, what happened? Photo bleaching means basically during the when the molecule is excited, some of the molecules may photo bleached. That is maybe chemically, or the activity of the molecules maybe destroyed in some to some extent, and due to the interaction with the radiations. But we need to choose in such a way so that there is a high quantum yield of the donors and the acceptors, especially the donors. So otherwise there is a chance for photo bleaching, and photo bleaching, if there is a photo bleaching, so the freight efficiency will be decreased, because it is the essential or to when the molecule is in the excited state, though, so the efficiently the electron, the atom molecule should transfer the energy. But if it is photo bleach, so it will be very problematic for the choosing the wavelength, choosing the appropriate conditions is important, so that we can minimize the photo bleaching for the study. Okay.
Actually, Dr. Joshimuddin, he asked, instead of L-Cysteine, whether any other aromatic amino acid be used in this process?
Yes, we have checked. No, yes, that can be done. But I have shown the experiment for the L-Cysteine only. But you can design other sensor for which the active sensor molecule can interact with that selectively with that amino acid. But it depends upon the interacting group or the moieties present in the amino acid. So it may be possible to design other but in this technique we have checked several other amino acids, other 19 amino acids. We did not find in such interference of the other amino acids. But definitely you can design some other materials where there is a selective interaction with the other amino acids. So it's not a problem. Thank you.
Okay, actually, Dr. Joshimuddin make a comment. not DNA, but hydrocarbon functions are the building blocks of primitive cell, sir. Actually, I think it is not a question, it is a comment, but I am requesting you to whether this comment is justified or not. Please.
Can you please repeat once? Can you please repeat once again?
Not DNA, but hydrocarbon functions are the building blocks of primitive cell.
Oh, yeah, definitely. No, I was talking about that. The DNA is the basically, if you talk about the cell, that functions the basic character of the cell basically. I mean, it contains the genetic the material. So that's why I'm talking about it is the primitive in that sense actually. So it functions, the DNA functions the basic character of the cell, and which function, and cell is basically containing the biological or physiological processes. That's why I have mentioned that as a primitive in that sense only. But that is true, that is the proteins are basically, and amino acids are the basic building blocks for the cell. Okay.
Okay, dear participants, any question from your side? So I can't see any more question in the chat box. So if no more question, then I can conclude this session. Any question from your side? So no question, then thank you, Dr. Pal, for your nice presentation, and thank you all the participants for your patience to listen the lecture.
Thank you, Surya sir, for nicely conducting the sessions, and also thanks to the speaker, Dr. Pal, from the organizing committee and from the departments. There is a small announcement. Now we shall share a feedback link, and this is different than the assessment link. So please fill up the form and submit as early as possible. And next session will start at 2 p.m. and after the end of the session at 3:30, we shall arrange a small valedictory session, and I request all of you to attend the last session as well as the valedictory session. Okay, thank you very much.